This is Chapter 11C, or Part 3, Fundamentals of the Nervous System and Nervous Tissue. The synapse is the area where a neuron is going to be communicating to another neuron or to an effector cell. The effector cell is either a muscle or a gland. Because the nervous system, remember, is a communication system, it has to take that information that it has received and pass it on to the next player in the role, if you will. So where it's passing it on, that communication from one neuron to what it has to communicate it to, the information to, is the synapse. The presynaptic neuron, that is the one that has the information and has to send it. The postsynaptic neuron is the one that's receiving the information from the presynaptic. Now, understand that neurons can act as both. So you can have a sensory neuron that is a postsynaptic neuron when it's receiving information from a receptor. It's getting that stimulus information. So in that case, it's a postsynaptic. But then it becomes the presynaptic neuron when it has to send that information to the interneuron or the association neuron, whichever you wish to call it, that's in the central nervous system. So that interneuron becomes the postsynaptic neuron. Then it becomes the presynaptic neuron when it takes that information and has to send it to the motor neuron, which is the postsynaptic. So you can play both roles depending where you are in terms of the pathway of the impulse being sent. And this is a really cool picture. You know, I just have to affirm to you guys sometimes that, yeah, I'm a science nerd. I see a picture like this and my first thought is, oh, look how cool that is. And I wonder how long it took him to get that picture. But you can see right here, I think it's cool just to see the axon itself. And then here's another axon over here. This yellowish area here, this is the soma or the cell body of another neuron. In this case, it would be the postsynaptic neuron. So an impulse that's going to be coming down this axon. So this would be the presynaptic. It's sending the impulse this direction, and it's going to send it to this neuron right here. The synapse is, think of it, it's the connection point. See how the axon terminus is right here? It ends at the soma. Here's another synapse right here. And it's just like, oh, isn't that really cool to be able to see that? The synaptic connections, sometimes you have an axon connecting to a dendrite, and then it would be called an axodendritic connection. If it's an axon with a soma, just like that previous picture that we saw, it's axosomatic. So we give it different terms, and you can look at it, the term, the name, to figure out what is connecting with what. Axonal is axon to axon. If you have a dendrite to dendrite, dendrodendritic. Somatic dendritic is dendrite to soma. So it can connect in multiple places. Excuse me. There's two different main types of synapses, a chemical synapse and an electrical synapse, which we'll see in just a moment. Here's another really cool picture, a schematic drawing of it though, showing here the axon connecting with the soma, so it's axosomatic. Axodendritic, here you have an axon synapsing with dendrites. And axonal, you have an axon stimulating right here the synapse with the beginning of an axon, because this is the axon going this way. So chemical synapse, this is the most common of what we see, where what's going to happen at that synapse is that you have your presynaptic neuron is going to be releasing a neurotransmitter, which is a chemical, which is why it's called a chemical synapse. Usually there's a couple different parts of what's going to happen. You have the axon terminus of the presynaptic neuron. It is going to be containing these vesicles known as synaptic vesicles. And those vesicles are filled with the neurotransmitter. That's where it's at. So it's inside these vesicles, which are inside the end of the axon terminus is the end of the axon. So we're talking about the presynaptic. So that's the first part of it. The second part is the receptor region. That's going to be on the postsynaptic neuron side. And that's going to be the receptor that's going to be very specific for that particular neurotransmitter. And remember, the presynaptic neuron and the postsynaptic neuron, or the effector of, say, the muscle, they're not physically touching each other. There is a little tiny space between them that's known as the synaptic cleft. So they're not physically touching. They're really, really close. It's kind of like when you were a kid and your sibling was bothering you and you wanted to bug them and you're like, I'm not touching you, I'm not touching you, but you get as close as you can, but you're still not touching them. That's kind of what it's like. An electrical impulse is going to come down the axon. It's going to switch, become chemical, and then you're going to, on the postsynaptic side, generate electrical impulse again. So how are you going to move things across that synaptic cleft? Well, you can't have that electrical charge go directly from one presynaptic neuron to the postsynaptic because you've got the space there. So that's why you have to have this chemical event here. You are going to have to release the neurotransmitter. It's going to diffuse across that synaptic cleft, and then it is going to bind to the receptor on the other side. That's very specific for it. This allows for the neurotransmitter to move in one direction because diffusion states things are going to flow from high concentration to low concentration. So if you release it on one side, it's very high concentrated there, so it will just naturally diffuse towards the postsynaptic side. There are basically 6 steps where it's been divided into 6 steps to explain how all of this works. You have an action potential that's arriving at the axon terminus. We're on the presynaptic side. So you had whatever stimulus it was, the action potential, you had a graded potential, it generated the action potential, it's running down that axon, it gets to the axon terminus. This change, because remember, even in the axon, it's negative on the inside. Well, that action potential is coming down the axon and it's changing it to positive. So you've got a change in that electrical charge, and that's what opens your voltage gates. Well, you have calcium voltage gates that are normally closed because of this change in the charge, which is the action potential. It triggers those gates to open. Calcium is normally high outside the cell, but very low inside the cell. So when you open those voltage gates, how did you open them? Because of the action potential coming down, you're going to have an influx of calcium. It's going to flow into the axon. And that is what this is showing here. This is your presynaptic neuron. Here's the axon. Here's the end of the axon. You tend to have these little swelling bulbs, if you will, on the very end, on the terminus. So the arrows here are showing the action potential, that change in the charge coming down. These are your calcium voltage gates. So the change in charge hits them, it opens. The calcium floods in. These are your vesicles, your synaptic vesicles. The little green dots are showing the neurotransmitter that are stored in there. What we see next is that influx of calcium flows in, and it's actually going to bind to certain proteins, and it's going to trigger the release of that neurotransmitter. So the calcium, basically, it binds to this protein that's going to react. It's a cascade series of steps, so one protein binds to another. Bottom line is what it does. That calcium flows in, and it's going to trigger those synaptic vesicles to move and fuse with the axon membrane. And when they do that, by exocytosis, they're going to dump all of that neurotransmitter into the synaptic cleft area. The higher the impulse frequency, the more vesicles are going to fuse with the membrane, leading to more neurotransmitter being basically dumped out of the cell. So here are the vesicles moving down, and you can see the little green dots down here. That's the neurotransmitter that's being dumped out of the cell. High concentration on the presynaptic side. This whole area is your synaptic cleft. So now the neurotransmitter is going to diffuse across. These purple structures over here are the receptors that are very specific for the neurotransmitter to bind. So that neurotransmitter diffuses across. It binds to that receptor. It's the only thing that's going to bind to it. So that's a chemical gated ion channel. So where you have the binding of a chemical, in this case it's a neurotransmitter. Remember, whenever you have two things bind, it causes a shape change, and that's going to open the channel. When you open the channel, typically these are sodium. Sodium is higher outside the cell than inside the cell. So the binding of the neurotransmitter causes a shape change and opens these gates, these chemical gates, allowing sodium to move in to the cell. So that's what you're seeing here. It's going to bind, open those gates. You can see right there, it's very specific for it. You have an influx now because you have sodium flowing in. Sodium is a positive charge, higher concentration outside the cell than inside. So when you open that gate, yes, it's going to flow in. Inside the cell is slightly negative, as we've already talked about. So when you have sodium, which is a +1 charge flowing in, that is going to change right around this gate area to a plus charge. When you change the charge in a very local, small area here, that is a graded potential. So you are creating a graded potential. You are changing that potential, that electrical charge inside the cell, just in a small area. Well, how do you stop all this? How do you close that gate? Whatever happens to the neurotransmitter? Well, that's going to be one of the last steps. The neurotransmitter that's binding to the receptor, as long as it's bound there, the gate's open and you continue to have the graded potential. So you have to be able to regulate that. You have to have a way of stopping it. Usually within just a few milliseconds, what's going to happen is one of three things. Either that neurotransmitter is going to be taken back up by the astrocytes or by the axon terminal itself. Two, it can be degraded by enzymes. Acetylcholine is broken down by acetylcholine esterase. Or three, it can diffuse out of that synaptic cleft. And that's what it's showing over here. It can be taken back up. It can be broken down, or it can just diffuse out in here and eventually be broken down. When it is removed from the receptor, by whichever one of these ways, it's going to close the gate. Because remember, when you bind two things together, you get a shape change. That's what's opened the gate. But when you remove it, when you unbind, break a bond between two things, you usually get a shape change. And that's what's going to close it back to its original closed position. Just FYI, there are different diseases and disorders that cause changes in the presynaptic and the postsynaptic neurons, which is one reason why it's very important to understand what the normal physiological processes are, so that then you can recognize when something's not working the way it's supposed to. As an example, there are two microorganisms. They are in the same genus but different species. And one of them causes the disease botulism, which can be fatal, a very, very serious disease. It is caused by a bacteria, and the problem is the bacteria produces a toxin, and it's the toxin that causes the problem. What the toxin does is it interferes with the binding of calcium. You still have your action potential coming down the axon terminus. It opens the calcium gates, but the botulism toxin is going to interfere with the calcium binding to those proteins that are supposed to then trigger the release of the neurotransmitter. It interferes with the step. It allows calcium in, but it interferes with it binding with what's known as the snare proteins. So what happens is you never get the release of the neurotransmitter. If you never have the release of the neurotransmitter, obviously it never binds on this side, on the postsynaptic neuron, or on the muscle. If you never have the binding of acetylcholine on the muscle, you will never get a muscle contraction. Now, in terms of cosmetics, they've used that toxin in such a way, if you never get the muscle contraction, then that means you never get contraction, frowns, and all of that. My caution on Botox treatments is that this toxin, it is naturally produced by the bacteria. It is the most toxic chemical naturally produced known to man. It can kill you. If it is in the wrong place, it will kill you. I'm just letting you know this to be very careful. If you choose to get Botox injections, make sure the person who is injecting it knows what they are doing. It causes a form of paralysis because you never get the muscle contraction. The other situation I was talking about is also caused by a bacteria that produces a toxin. As I said, it's the same genus, different species, and the toxin I'm talking about is the tetanus toxin. In this case, what happens with this bacteria is that the toxin that it produces, it interferes with acetylcholine esterase, the enzyme that's supposed to be breaking down the neurotransmitter. So if you never break it down, it remains attached like over here, which means this gate is always open, which means you always have a graded potential and you can't stop the muscle contraction. So with botulism, you never get the muscle contraction. Tetanus is the opposite, where the muscle stays in a constant state of contraction and it will never relax, both of which can be deadly. So once again, I'm mentioning that from the standpoint that to understand in those cases, for me personally as a microbiologist, which is my specialty, I love studying about how the microorganisms work. How do they cause diseases? Well, for me to understand how those disease processes work, how does it cause paralysis? Well, it's interfering at the neuromuscular junction. Well, how is it interfering there? Well, for tetanus, it's interfering with acetylcholine esterase. It makes sense when you know what the normal functioning is, so you can see what steps are being interfered with, and then how you get the disease symptoms from that. It just is another piece of the puzzle. Synaptic delay just refers to the time that's needed for that neurotransmitter to be released, diffuse across the synapse, and then bind to the receptors. There is a delay there. The action potential travels down the axon very, very quickly, but it's going to be slowed up in the synaptic delay, in that release of that neurotransmitter. Now, how much of a delay are we talking about? Well, it can take anywhere from 0.3 to 5 milliseconds, which I'm sure most people like me are going, that's not very long. But in the overall timing of things, that's the holdup. That's the slow, the step that kind of slows everything else up. So. The electrical synapses are less common than the chemical synapse. What happens here, typically it's when neurons are against neurons. They're in a situation where there are gap junctions, which are those connections, cell-to-cell connections, so you have to have adjacent cells. They almost look like little pores so that the cytoplasm can flow from one cell to the next. And so it allows the communication to be very, very fast. And whereas chemical synapse is unidirectional, this can be bidirectional. We do see this with some areas of like for eye movements, memory. It's fairly common in developing embryonic nervous tissue. The postsynaptic potentials, the neurotransmitter receptors, the ones that bind that neurotransmitter, there's going to be different things that play a role with the receptors. How long is the neurotransmitter remaining in the cleft? How much of the neurotransmitter is released and binds to those neurotransmitters? You end up with basically two different types of postsynaptic potentials. You can end up with what's known as Epsp, which is excitatory postsynaptic potential, and the other type is Ipsp, or inhibitory postsynaptic potential. The names, I know, sound really long and stuff, but let's just break it up, and it's fairly easy to see what's going to happen. Remember, the neurotransmitter binds to that receptor, and you tend to have the sodium, sometimes potassium, that are flowing. So you've got sodium flowing in, potassium flowing out. Now, the sodium tends to be flowing in a little bit faster than the potassium, so you end up with more positive charge coming in. That is going to cause depolarization. It's going to cause it to become more positive, and that's going to be excitatory. It's going to increase your chances of having a response, because the more positive you make it, the closer you're getting to that threshold of generating an action potential. Remember that threshold, that all or nothing. You reach that point, boom, you've got an action potential. So if you have a lot of positive charge coming in, you're getting closer to that threshold. So you're increasing the chances of having generation of an action potential. You're increasing the chances of, essentially, bottom line is generating a response. So if you look at it on a graph, here at minus 70 is the resting potential. Here's your stimulus. Here's the depolarization. This dotted line is that threshold. You reach that, boom, you are gone. You're going to zoom way up here to about plus 30. You're going to generate that action potential. Until you reach that minus 55 threshold area, this is just a graded potential, just in a localized area. So for an excitatory postsynaptic potential, EPSP, you're getting closer to that threshold. You're more positive. Now, for an inhibitory postsynaptic potential, what happens here is that you're going to typically trigger a chemical gate to open again because of a neurotransmitter binding. But in this case, instead of sodium, usually it's either potassium and sometimes chlorine. If it's just a potassium gate that opens, remember, potassium is higher inside the cell than outside. So if you open a gate, where is it going to flow? High to low, which means that positive charge is going to flow out of the cell. If you remove that positive charge out, it's going to make the inside even more negative. Sometimes it may open up a chlorine ion gate. Chlorine is a negative one charge. Chlorine is higher outside the cell than inside. So if you open it, it's going to flow into the cell, which is also going to make the inside more negative. So if the neurotransmitter binds to a receptor, opens up chemical gates, and it causes the inside of the cell to become more negative, so it's moving even farther away from that threshold. It's causing, when you go more negative than resting, that's hyperpolarization. You're getting even more negative, and it's called inhibitory because you're reducing the chances of having any type of a response. Why? Because you're reducing and decreasing the possibility or inhibiting the possibility of generating an action potential. So if you look at it, you're resting here, here's your stimulus, you're going more negative. Threshold's way up here. You're going the wrong way. That's why it's inhibitory. You're making inside the cell more negative. Excuse me. Summation of the postsynaptic neuron, a single excitatory postsynaptic potential usually will not induce an action potential by itself. It has to be several of them and you add them together, or it's a summation. Now, there's two different types of summations, or two ways of adding things together, temporal or spatial. Temporal is when you're getting in sequential order, very rapid fire order. So the first one produces an excitatory. Before it completely relaxes, you get a second one. You add it on to the first one. Then you add on the third one. You add them together. Spatial summation is when you have a large number of terminals that are being stimulated at the same time. So a lot of receptors are being activated, and you add them all together at one time. So here you have an excitatory response here, then later you're going to have another one right after, and then another one, and you add them together. Excuse me. When you graph that up, so here was your first one, it was starting to relax, not quite fully relaxed yet, you get the second stimulus, here you go. And then for spatial, you're adding them at the same time. You're getting these stimuli at the same time here. Both of these at the same time are stimulating these dendrites here. So is it one after another, or is it at the same time? And then this chart goes through and it's comparing the graded potential, which remember, it's a very localized area, versus the action potential, which is going to be a much longer distance. It can go down the entire axon. The graded potential is just going to be along the cell body or on the dendrites. Very short distance versus a long distance with the action potential. So this table goes through several different characteristics in comparing the graded potential versus the action potential. Here it looks at the initial effect of the stimulus. Notice here, this is much higher peak because you've reached threshold. If you don't reach threshold, you will never generate that action potential. Neurotransmitters, there's over 50 of them that are known at this point. The first one to be identified was acetylcholine, so that's the most well-studied one. Most neurons can make two or three different types of neurotransmitters. They have different receptors. Neurotransmitters are usually classified by their chemical structure and by their function. Like I said, acetylcholine was the first one identified as the one that is released at neuromuscular junctions. It's also released at some of your autonomic neurons and some central nervous system neurons. It is broken down by the enzyme acetylcholine esterase. Neurons usually function together in groups. That's what's known as neural integration. It allows for a more coordinated response, broader functioning, and obviously you have to have a lot of communication going on between them. The neuronal pool is the functional group of neurons, how they're going to be processing that information and sending that information on. In a simple neuronal pool, you have just a single presynaptic fiber, and it's going to branch and it's going to synapse with several different neurons. So it's just not a one-to-one, it's one to many. The discharge zone, those are the neurons who are closer to that incoming and fiber and are more likely to generate an impulse. Towards the edges of that, then you have your facilitated zone, where neurons, they're still being synapsed with that initial single presynaptic, but they're farther away. So they may not be stimulated unless there's another stimulus coming in as well. So here's your presynaptic fiber branches. So this area closest to it right here, this is your discharge zone. On the edges, which are further away, that would be facilitated. So these might not respond as quickly as these or as strongly. Serial processing, this is the patterns of neural processing. So you have the input traveling along one pathway to a specific destination. So neuron number 1 stimulates neuron number 2, which 2 stimulates 3, 3 stimulates 4. We see this oftentimes with spinal reflexes. Reflex is a very rapid automatic response to a stimuli. You always get the same type of response. You stick your hand on a hot burner, you're always going to move your hand away from the burner. We often refer to these as reflex arcs. There's 5 components. You have a receptor that picks up that stimulus. It will send that message to a sensory neuron. The sensory neuron sends it to the central nervous system, to an integration center with your interneurons or your association neurons, which is going to interpret the information. What do I need to do? Send the information to the motor neuron, which then activates the effector, which may be a muscle to move your hand. So it's these 5 different components. You can see that in the simple reflex arc, it's the serial processing, one-to-one. You get your stimulus, sensory stimulates interneuron stimulates motor neuron to the effector to get your response. Parallel processing is when you have input traveling along several different pathways. It allows for you to deal simultaneously with information to generate numerous types of responses. You often see this with higher level mental functioning, where you can get your, say, auditory, you're hearing something, but you're also visually seeing it. Maybe you're smelling something, maybe you're feeling hot or cold or whatever, and you're taking all of this information and processing it together. So you end up with different types of what we call circuits. It's how the neurons all basically connect to each other. And there's 4 types of circuits: diverging, converging, reverberating, or parallel after discharge. And this is the easiest when you look at it. So diverging, you have one input, but it's going to branch out and it amplifies it. So you have a single neuron that can activate, and oftentimes this might, let's say, be in the brain, where you can have a single neuron that has been stimulated, and it's now going to activate ultimately several different motor neurons in the spinal cord, which can then generate impulses to several different skeletal muscles to allow for a very well coordinated movement. I need to adjust my weight to pick up a book and also hold my coffee cup in my other hand while walking and watching where I'm going. And listening, in my case, listening, where's the cat, so it doesn't come running right out in front of me and trip me. So you've got all these different things going on. Converging is where you have several different inputs now, but only one output. So you're getting all of this sensory information in. And as the example says, you could get different sensory information and it's stimulating the same memory. Reverberating, the signal initially is going to travel down the chain of neurons, and then at multiple steps it can generate feedback so that you're always getting the stimulus. You have the one input here, but you get the same output to continue on. And then parallel after discharge circuit, this is where you have your signal coming in. It stimulates several different neurons in parallel, but then they're all going to converge here for a single discharge.